So in the last video, we covered respiration, focusing on aerobic respiration. Now let's talk about photosynthesis. For this unit, we have several learning objectives to define some words, talk about structures, diagram the flow of electrons, and then talk about the steps in some of these light-dependent and light-independent processes. Photosynthesis works to convert light energy into chemical energy, and it's used by our phototropes. And it involves two major processes. Again, the light-dependent capture of light energy that occurs in our thylakoids, and then the assimilation of carbon, turning carbon dioxide into carbohydrates, and this is considered a light-independent process. Now, if you look at the formula, it should look familiar to you as it's almost the reverse of respiration. And here we're going to use carbon dioxide to eventually make a sugar. These two reactions occur in our chloroplasts, which have a membranous structure, the thylakoids. The light harvesting is going to be done sequentially by photosystems here, and then eventually the light-independent process will occur in the space, the stroma, not in the membrane structure. And the light harvesting is done by systems called photosystems. And photosystems are going to use pigments to capture light and then pass that energy, those electrons, eventually to a reaction center where we have a pair of chlorophylls. And the chlorophylls are named by their optimal wavelength, either 680 or 700. Now I said that photosystems use pigments like chlorophyll to capture photons of light. Chlorophyll has a porphyrin ring structure that allows it to absorb the photon and then pass electrons through this ring. It is anchored into the membrane by its phylitol side chain. We also have other pigments, accessory pigments, things like carotenoids, which have other optimal wavelengths and can assist in light capture. So photosynthesis also uses a different electron carrier. So we talked about respiration, we talked about NAD plus and NADH for respiration. Very similar structurally, an extra phosphate group here. We get NADP plus and NADPH. Keep in mind P for photosynthesis. And shown here is the energy diagram. So photosynthesis is going to start with a non-cyclic flow of electrons. We start with our second photosystem. I apologize for the nomenclature. They were named in the order that they were discovered as opposed to the order in which they work. So we start with PS2, photosystem 2, and it's going to strip electrons from two molecules of water, pumping the hydrogens into the thylakoid lumen and releasing oxygen. Now, those electrons are going to be excited in that P680 chlorophyll pair, and they're going to bump up to a higher energy level. Those excited electrons are then passed along, eventually reaching our cytochrome complex, which is going to pump protons, even more protons, in the thylakoid lumen, and then we'll pass those electrons to our second photosystem, photosystem 1, PS1, to our second chlorophyll reaction pair, P700, which will again absorb photons of light and bump it to a higher energy state. Now, those electrons can then flow through and will eventually be used and transferred to reduce NADPH, our electron carrier. Like in the mitochondria, the movement of electrons and this proton gradient is coupled to the synthesis of ATP. ATP synthase is going to use that proton gradient that we make in our thylakoid lumen to, again, using a very similar structure, move that protons back down its gradient and use that energy to make an ATP molecules. Now, if the cell needs more ATP than it can generate just with those few NADPHs, it can then do cyclic flow of electrons. So rather than wasting the electrons on NADPH, it can pass them back to the cytochrome complex, which will use it to pump even more protons. Keep in mind that the protons are the thing that move through the ATP synthase, creating ATP. So it can pass them to the cytochrome complex, which will then pass them back to PS1, which will use photons to excite them, and it can go through this cyclic flow as many times as it needs to make ATP to meet its energy demands. Now, once the plant cell has used light to make energy, the cell can then use that energy to fix carbon dioxide and eventually make both starch and sucrose, our sugars. This light-independent process is called the Calvin cycle. And again, one of the products of the Calvin cycle will leave the cycle and form the backbone for sugars for the plant. The Calvin cycle also has three phases. In the first phase, carbon dioxide is added to 3 molecules of ribulose 1,5-bisphosphate, or RUBP, by an enzyme called Rubisca. And in the second phase, the 3 new carbon molecules are going to be reduced, gaining electrons from NADPH. Finally, we're going to regenerate our acceptor, our RUBP, and the cycle will also use some ATP molecules. This is the cycle shown in full. Again, carbon dioxide and water is going to come in. We're going to use our electron carrier, NADPH, and some of the ATP that we made earlier. So this video, we covered the process of photosynthesis. And in the next video, we're going to cover cell signal transduction